Okay, we are live. And again, please interrupt with questions anytime. If you recall, last week we kind of ended on a bit of a cliffhanger. In the last little bit of lesson 3, we were looking at how you would use a additive model, a GAM, to account for any spatial trend in environmental factors that occurs over your study area that cannot be accounted for by the random effects of blocks and by the fixed effect of treatment. We looked at different ways of accounting for that. We looked at the residual covariance structure way, and now we were looking at the additive model way. But all we had managed to do when time ran out last week was to fit just a regular linear mixed model to the data that did not account for space. So now we're going to account for space. So let me do my screen share here so I can remind you where we were, and then we'll all log into SciNet together and pick up where we left off, and I'll help you run the code to get you to where we were last time. Oops. Here we go. Okay, so this is the slide just to remind you what the example data set is. Again, it was a field trial of barley being grown in Scotland, and it was a trial of dozens of genotypes, and each genotype was planted with and without fungicide, and as a split plot, and with fungicide being randomized at a larger level than individual little plots for the genotypes. And so there's eight of these large plots, and the whole, because the whole thing was replicated 4 times and yield was measured. And if you recall, we made a bunch of maps showing that even if you accounted for block and plot and the different treatments, there was still some residual variation that had a spatial pattern. And if we have a spatial pattern in the residuals, or a pattern of any kind in the residuals aren't supposed to have a pattern. They're supposed to be random noise. And so any pattern we have in there is something that is a red flag that we should have put that term into the model. Okay, so let's go ahead and go back. Everyone, if you're running it on your own computer, open up RStudio. If not, you will go back to SciNet and log in. I already have my session started, but if you go to the interactive sessions, my interactive sessions, once you're logged into SciNet, if you remember, you can just click on this refresher icon from last week's session if you still have it there, and then it will immediately start a new session with the exact same parameters as last week. But if you don't have that, you'll just have to go to the RStudio server launcher window and start a session with the same the parameters. We have to start it under this workshop account and series R4.4. 4 hours, 4 cores, 16 gigs, and we'll use the Bayes 2025 reservation, and then you can launch that. And then you should be able to go into the RStudio server. So can everyone give me a thumbs up if you've either got that or are on the way to getting there? Okay, excellent. And of course, you can get help from me or Grant if you cannot make it there. So now I've logged back in to the RStudio server, and my Bayes 2025 project is still open from last time. If you don't have that, if it doesn't have that project, you'll have to go up to this upper right and open the project. Bayes 2025 should be on your recent list. And the worksheet from lesson 3 that we've been working on filling in is still there from last time as well. But just to make sure, I would recommend going into the Bayes 2025 directory, opening up the worksheets that are already filled in, the worksheets filled in directory, and open up the filled in worksheet for lesson 3, because that will just verify that that we'll have everything just so in case you didn't type in the code, part of the code or anything from last time. So what I'm going to do is I'm going to load all the packages by going all the way to the top of that worksheet, and I'm going to run everything up to the options line on 29. I'm gonna hit Control Enter to highlight everything up to line 29 and hit Control Enter. And that will load all the packages and set the options that we need to do what we're going to do. And then you can scroll down past the cow example that we did last time, all the way down to well, it might be a different line number, but under the heading of spatial additive model. And then I'll put my cursor on line 122 and hit Control Enter there to load the Durbin et al. Split plot data set into my workspace. And just to, we can just go ahead and look at all the maps again that we made last time, just to remind you, since it's been a whole week since we last did this. So I'm going to run the code to draw each of these maps so you can be reminded what we were dealing with. Yeah, can everyone either give me a thumbs up if you're have made it to this point, or if you need help, please let me know. Okay, it seems like people are able to get caught up again, so that's good. Okay, so I'm just gonna draw these maps again. Feel free to follow along on your own, or just look at what I'm doing. So I'm gonna hit Control Enter to run this first map. Code to make the first map that just shows there's 4 blocks all in a row. And for some reason, the X coordinates of the plots are the beds, and the Y coordinates are rows. So they're like little square plots. Each combination of bed and row is an XY coordinate. I'll hit Control Enter again. And you can see that the randomization of fungicide was done at the main plot level. There's eight of those total. Next, I'll run another line of code, the next map, just to show that each plot is divided up into, well, each main plot is divided into 70 subplots, one for each genotype. And lastly, and here's another map showing how the yield varies over space. And there you can see that there's clearly a difference in between the different fungicide treatments. You can kind of see where one main plot ends and the other begins, but also there's kind of like some hot spots of high and low yield that have nothing to do with any of the treatments. Those are just natural variation in yield that occurs because it's not a completely homogeneous area. And we can see the spatial pattern by randomizing the location of the subplots within each plot. Just hypothetically, what if they were shuffled? Would the pattern look more random? If it does, that means there's kind of like some pattern in the data as it is. I'm just going to keep running this code. I'll set my random seed. I'll shuffle up the X and Y coordinates, or I'll shuffle the yield values with respect to the X and Y coordinates within each main plot. And then I'll plot those shuffled values just for fun. And you still see the difference between the main plots, the fungicide treatments, but we don't see those hot spots on the north and south anymore because they are not related to block. Those are just spatial variation. And then this is still rerunning code from last week. The next thing is that we fit a linear mixed model that accounts for all the random effects according to the experimental design, but does not account for any of that spatial variation yet. So the response variable was scale parentheses yield, which is basically just a Z transformation on yield, just to make yield be in standard deviation units so that we don't have to consider the actual units of yield when we define our prior distribution on the fixed effects. And we have our genotype, fungicide, and their interaction as the fixed effects. And we have a random intercept for every block. There's 4 of them, and a random intercept for every combination of fungicide and block. Those are the main plots. There's 8 of those. And we've put a prior with three as a standard deviation on the fixed effects, because again, we have our response variable is in standard deviation units, so a 3 standard deviation change is pretty big, so that's a conservative prior. So I'll hit Control Enter to load the pre-fitted model object there. And then we said, okay, let's plot the residuals of that model to see what they look like. Let's see if there's a spatial pattern just by eyeballing it. So we'll run this line to generate the residuals. And then we'll add a column to the data with the residuals of the model that ignores space. We'll plot them. And we see that now that the treatments and genotypes have been filtered out, the spatial pattern is kind of even more obvious, because any pattern that's left in the residuals is spatial variation that exists outside of treatment in the data. You can see there's like some areas of low yield, like maybe in the top middle, and areas of high yield, like in the the southeast corner. That's our so we say, well, let's account for that. And the last thing we did last week was that you can also calculate a formal statistic quantifying how much spatial pattern, how much spatial autocorrelation there is in these residuals. It's called Moran's I. It's basically the correlation between how close together 2 residuals are and the inverse distance between the 2 residuals. So the higher that correlation, the more spatial pattern there is. The more likely 2 close together places in space are to have the same similar residuals, like the first law of geography. So I'll run this line to generate the inverse distance matrix, set the diagonal to 0, and calculate the Moran's I statistic, which is, it's not super high in an absolute sense. It's 0.04. It can go all the way up to 1 if there was perfect correlation, but it's still non-zero, and there is a spatial pattern. We can detect it, and so we want to account for that in our statistical model. Okay, everyone back up to speed with where we were last week. Give me a thumbs up if you're following. Super. Okay. Now I will I'll take away this cheat sheet here, and we'll go back to the worksheet that, hold on one second. We'll go back to the worksheet that is not filled in and has blanks in it. And what we're going to do is we're going to fit these generalized additive models. We talked about how they basically do what's called a smoother or a spline. They fit a combination of locally weighted polynomial functions to kind of fit a wiggly surface that would capture all the little local ups and downs in the spatial structure of the data. Like it would have a low value in this area where it's low, and a high value here where it's high. And there is this parameter K, which determines how wiggly and squiggly this surface can get, how closely it can fit the little individual spatial variation in the data. But the good thing is that GAM's generalized additive model, this implementation of them has its own built-in penalizing term that will keep it from overfitting the data. It will fit kind of the important stuff and not fit the random noise. And so the higher but this parameter k, the higher we set it, the more squiggly it can get. But because of the fact that it's not overfitting, we can actually set k to a pretty high value without overfitting. But I'm gonna try out different k values, which are basically the number of points in a spatial grid that would be overlaid over the number of points that the surface can have in a grid. So the more points, the more ups and downs it can have. So how would we code that? Well, the code for this GAM is pretty much exactly the same for the most part as the code we were using for the generalized linear model. We've got our same response variable, our same fixed effects, and our same random effects, same prior on the fixed effects. The only difference is this plus S part. S is the function used for the spline, the smoother, and we're going to fill in the arguments of S. Now, because this is two-dimensional data, right? It's spatial data. It's got an X and a Y. We need to specify the which columns in the data are the X and Y coordinates that we're fitting the spatial spline over. And you remember the X column is called bed, comma. The Y column is called row. Those are the names of the columns in the Durbin split plot data set that represent the coordinates. And we will add the parameter k, and we're going to start with k equals 10. Let's see how that does. And then the next argument is BS equals, quote, GP. What does that mean? The BS stands for basis, and the GP stands for Gaussian process. That is a specific kind of process of mathematical function that we are using to model the variation in space. And Gaussian process is probably the most flexible one, and it works very well in most circumstances. The only reason you might consider other ones for spatial data is that Gaussian process is very computationally intensive and it does not scale well to huge data sets. But because we have a relatively small data set, right, there's only it's only like a 10 by 50. There's only around 500. Well, there's 560 data points. That's not that many. It's fine to fit a Gaussian process spatial additive term to this data set. I'm not going to go into the other options right now. I'm also not really an expert in it. So if you have some big data set that you're interested in fitting GAMs to, we can talk about other options. But for now, let's just say Gaussian process is good for our purposes. So we can, yep, that's all blank that I left. Other than that, everything is the same. We're doing 4 Markov chains, 4,000 iterations, each of which the first 2,000 are going to be warm up. And I'll hit Control Enter to run that or load the pre-fitted model object. Whoops. Apparently, I have done something wrong because it's starting to rerun the model. So it shouldn't do that. So let's see, what did I do differently? Let's see if the 25 one works. Yep, maybe I forgot to set the, let me just make sure that I set the options for model refitting to not automatically refit the model. So I'll go back up to the top. I'm going to run this line again, line 29, just to make sure. Let's try that again. Is anyone else getting it to load the pre-fitted object, or is it Yep, so it's starting to, yep, I see. Let's see. Let me check that. I'm not sure why it's doing that. Did we skip any let's see, did we skip anything on how the data was processed before we before we fit the model? No, I don't think so. Okay, so let's see. Well, go ahead and stop. If you have it starting to compile, just go ahead and stop that. Let's try options. I'll type this in here because I don't want to sit here and let the models refit. So we're going to do options brms.file_refit equals quote never. So that will hopefully force it to not refit the model. I'll hit Control Enter to run that. Now I'll run that. Okay, so I don't know why it detected a change in either the model or the data and was trying to refit the model. Yes, thank you, Grant. So that will kind of force it to not do that. Yeah, just stop it if it's starting to compile and resample the model, and then set this option to not refit. That will force it to just load it from the pre-fitted model object. And then we'll go ahead and also run the same exact code, except we're going to set k equals 25. And also we're doing k equals 50 as well. The code is exactly the same for the three models, just one's 10, one's 25, and one's 50. Again, the higher the k, the more finely we can capture spatial trends in the data at the expense of maybe it's more computationally intensive. So I've run the k equals 10. I'll put my cursor somewhere in here, and I'll run the k equals 25, and I'll run the k equals 50.
Sorry to interrupt, Quentin. Can you
Go ahead.
Scroll back up to that options line? I didn't quite get the entire.
Yes, Grant put it in the chat as well.
Oh, sorry, I
so
missed
you
that.
can
Never
see it
mind
there.
then.
Yep, no worries. Yep. Please give me a thumbs up if you're able to get those to load and as intended. Okay, many of you are good. Excellent. Now, let's look at, out of the four models we fit so far, we fit one that ignores the spatial variability, doesn't account for space. And we fit three different GAMs with progressively increasing values of k. Let's do what we have done in previous lessons and do the leave one out LOO cross validation to see which one of them best fits the data, or yes, is a best fit to the data. So we're going to use this function add_criterion to add the information criterion to each model, and then assign the result back to the model so that the information criterion is stored in the model object. And in quotes, Lou, that's the name of the criterion that we're going to add. And then we'll highlight all four of those. So we're going to do the exact same thing for each of the four models and hit Control Enter to run them. And because the criteria are already stored in the pre-fitted model object, it will run quickly. And then we're going to compare, now that we have a pre-computed information criterion based on leave one out cross validation for each of the models, we can compare them Lou_compare and set each of the four models' names as the arguments. And then we'll hit Control Enter on that. So we can see the best fitting model is at the top and the worst at the bottom. And the differences in the information criteria based on this ELPD, expected log pointwise posterior density, is very large. And especially bad is the original model that we fit that ignored space. So you can see that actually it seems like we've set k, we might even try, you could even try a higher k, because it seems like the higher we set k, the better the fit. So we actually could maybe even try a higher number than 50. I'm not going to do it for right here in this this lesson, but feel free to play with that on your own time and see if you can get a better fit. Yes, Brad, do you have a question?
Yeah. So is there kind of a cutoff that you should use for that, where even if your K was lower, but the difference between those two models is only, like 3 points, is
Right.
it
So
still
yes.
worthwhile to set that K higher?
Yeah, I think eventually the higher you set K, the difference would flatten off and there wouldn't be any meaningful difference between the models, because of the regularization that is being imposed to prevent it from fitting it too closely. Eventually, as you increase K, the model would not differ at all from, they would just end up wouldn't be different. So yeah, I mean, I would say, if the standard error of the difference is bigger than the difference itself, then it's not a meaningful difference. So that's what I would go off of. Actually, there
Okay.
is a formula you can use to translate this into like something akin to a p-value if you wanted it in those terms. I don't know it off the top of my head, but I can pull that up for you if you're interested. You can actually get a sort of p-value like statistic for the difference between these two models based on this ELPD.
Right. Okay. I was just wondering,
Yeah.
I didn't know if there was
Yeah, for sure.
A difference in ELPD by 3 units may not really mean that much. But
Yeah, it doesn't. Yeah, I would say not. But yeah, I would just look at the standard error of the difference and see,
Okay.
like, the difference is 60, standard error of the difference is only 9. So it's like, well, that's a large difference relative to the uncertainty in it.
Yeah.
Yeah.
Okay.
Cool. So now we say, okay, well, K equals 50, at least among the models we tried, is the best fit. So let's use that model to make our predictions. But I also wanted to do a couple of diagnostics so we can see how it improved. Like, if you remember, the plot that we still have up here is the residuals from the model that ignored space. So let's get the residuals from this K50 model and let's plot them. Let's see if we can discern any spatial trend in those residuals, right? Like, the residuals are the random noise left over after the model. So now these residuals are going to be the random noise that's left over after we've accounted for fixed effects and random effects and the spatial trend. So hopefully there should be really no pattern in these residuals that we can see visually. So I'm going to use mutate to add a new column to our original data. I'm going to call it resid spatial, residuals of the spatially aware model. And I'm going to use the function residuals to extract those residuals from the model object, which is called Durbin underscore GAMM for generalized additive mixed model underscore K50. And we're just taking the first column because we're just getting the posterior median of the residuals. We don't really care about the uncertainty in the residuals just for the purpose of making this quick map. So we're just taking the first column. I'll hit Control Enter to run that. And then the next lines of code is the same code we've been using to draw the map, every other map, except the only thing we're going to change is the fill variable is going to be this new column, resid underscore spatial is what I named it just now. So that's what I'm going to type in there. I'm going to hit Control Enter to run that. Okay, so now take a look at these beautiful residuals. It looks just like static, it's just noise, right? There's not, I mean, maybe if you squint, you could see something, but there's really very little space. There's no large areas of high values or large areas of low values. It's pretty much looks random, especially if I would go back one to look at the previous plot of the residuals of the other model. You see those clear spatial structure. Here's a low spot, here's a high spot. Back to these ones, maybe they're much smaller areas of spatial patterning. It's much less obvious. And we can remember we calculated this spatial autocorrelation statistic for the residuals of the previous model and found that it was 0.04. and it had a very low p-value associated with it. Well, let's do it. Let's calculate that I statistic for the residuals of this model. So which column are we calculating the statistic for? It's this resid underscore spatial, right? So in R, if you say a name of a data frame, $, the name of the column, it'll pull that column from the data frame. And we already created an inverse distance matrix for every pair of plots in our data set. Last time when we did the I statistic, which we called Durbin underscore inv underscore dist. So the Moran's I takes two arguments. The first is the value we want to see if it's spatially correlated. Those are the residuals. The second is the inverse distance matrix between all the pairs of residuals, their distance in space. Now it's going to say, is their distance in space correlated to their distance in how different they are? Control Enter. So now it's negative 0.005. It's, the absolute value of that is very, very close to 0. And it even has a p-value that is shows that it does not differ from the null expectation if it truly was spatially random. So this is great. We've sort of successfully accounted for this spatial autocorrelation in the spatial patterning, the spatial variability that existed in this field where the experiment was done, which will now give us a better answer of the things we care about, which is the differences between the effect of genotype and fungicide. So now you may wonder, okay, we went through all that. Did it actually have any discernible impact on the inference we would draw from this data? Like, what if it didn't really change the bottom line? What's the point of going through that complicated procedure of fitting a GAM that's very computationally intensive and so forth? So I haven't really done a fill in the blank here, but I'll just explain. This is just as a proof of concept. I've calculated the estimated marginal means for the model that ignores space and the GAM model that accounts for space for each combination of genotype and fungicide. So that's going to be 140 values, right? Because we had 70 genotypes with and without fungicide. That's 140. And then I've just kind of thrown them together into a data frame and labeled them as non-spatial and spatial. And then I've written some code to plot that so we can compare them. So let's run this code to calculate the means and stick them into a data frame together. And then let's plot them and see what does it look like. So there's two fungicide treatments, F1 and F2. And and the orange is the model that ignored space, and the blue is the model that accounts for space. So, the difference is not enormous between the two models, but, you can see that these orange points, especially for treatment fungicide 2, are systematically higher for a given genotype than the corresponding blue points from the spatial model. So maybe we could say, well, if we ignored the effect of space, we might be slightly overestimating how beneficial fungicide 2 is for barley yield. So it's like that could be a consequential inference. You don't want to say that the difference between two fungicides is this when part of that difference is actually just due to spatial variability that existed in the particular field where you did your experiment. So we can sleep better at night knowing that we're not trying to bamboozle people by ascribing some of the difference in fungicide to, or ascribing all of that difference to the fungicide effect when some of it is actually just due to spatial variability. So this model the spatial model has slightly lower overall predictions for fungicide 2. So yeah, there's a bit of an effect of doing this. Give me a thumbs up if you're following me at least somewhat. Cool. And of course, please interrupt if you have a question. And before we move on to other things, I have a few more just proof of concept things so you can kind of see what this GAM model is doing. What I'm doing in the next bit of code here is basically creating a prediction for all the spatial coordinates and averaging it over the treatments and over the random effects so that you can see what the actual spatial field that is predicted by, that is fitted by the GAM model actually looks like. And yeah, I've just done that by averaging over a few draws, 10 draws from the posterior, just to get one value for each coordinate to plot on the map. So yeah, I'll just run all that code. Don't worry too much about the ins and outs of that code. This is just as a proof of concept. So we're generating a prediction for every coordinate averaged over the treatments, and this is what you get. So here you can see the shading color is the value of that spatial smoother at a particular point in space. So the whiter areas are areas where it is more negative, and the redder areas are the areas where it's more positive. So if you think back, this spatial smoother actually resembles the residual plot that we made of the non-spatial model. So it's lower in this area where we found the yield was a bit lower than we would expect, and the spatial smoother is a positive value over here on the right in that area where yield was a little bit higher than we would expect. So this smoother is actually like taking those residuals and fitting kind of this smoothed function to them. And that's why the residuals of the spatial model don't have that pattern, because it's accounted for by this spatial smoother that we've added to the model. So I thought that was just a nice way of illustrating that. Okay. Lastly, before we go on to lesson 4, again, I wanted to show you and describe to you how you would actually present all this stuff in a manuscript. So I have in the slides and also in the full text version of the lesson. I'll go to those slides real quick. We did all of this. Yes, how would we describe it in the methods? And I've just given you a table. I haven't done the full results write-up and figure as I've done for some of the other stuff, but for your own reference, you may be interested in how you would write up everything in the methods. And again, because I usually think that all this accounting for space is not really part of the objective of your study, I would put kind of the model fitting and model comparison results in the methods section and not necessarily in the results of the manuscript. But, that's a stylistic preference. Yeah, again, you would talk about how you tried out different basis dimensions and compared them with leave one out cross-validation, and then explain why you arrived at the model that you did. If you tried out different models, it's good to say in the methods everything that you tried out instead of just, and why you picked the one you did, instead of just telling the last and best model that you did and acting like that was the one you intended to use all along. It's more honest to explain the selection process, and it helps you be kind of more explicit about it. And lastly, yes, what we need to do to get data for our manuscript quality table would be we now might want to know within each genotype, what is the effect of fungicide on yield? And the way we would do that is by using the formula within the EM means function, the formula pairwise tilde fungicide type genotype, and that's going to give us a mean for fungicide 1 and fungicide 2 within each genotype, and then it's going to take the difference between each of those means. So what is the relative difference between the fungicides within each genotype as estimated by the model. And again, these estimated marginal means are going to average over all that spatial terms and all the and everything like that, and give us the overall predictions. I'll hit Control Enter to run that. That's by genotype. And then overall, if we wanted the fungicide effect averaged over all genotypes, we would just do pairwise by funge for fungicide, and that will give us one overall effect for fungicide. Yes, and we can run those lines. And again, it states the results may be misleading due to involvement in interactions. Again, yes, we are looking at the main effect of fungicide, but it's averaging over all the genotypes. Maybe some genotypes have a positive effect of fungicide 1, some have a negative. If you just give the overall, if there's a strong interaction, that may not be supported by the data. Yeah, so I'll leave looking at the manuscript quality tables and everything to you as an exercise, and playing with the estimated marginal means. You can feel free to do that on your own time and explore that and ask me if you have any questions. Yeah, so that concludes the spatial additive models part of what we're doing. Does anyone have any questions for me? Okay, so let's take a 5-minute break, and if anyone has any questions during that time, please ask me, and then we'll start on the next part of the content at 2:50 Eastern, so 5 minutes from now. So yeah, take a quick break. And I'll get everything set up for the next lesson. Okay, we'll get started back again in just a minute. Okay. All right, so I'm go ahead and continue on with lesson 4 of the Bayes workshop. So again, this is new content that I haven't taught off of yet. So if you notice any typos or anything that looks off, I'd love to hear about it. What are we going to be talking about for the rest of today? As you know, we're working our way through this BRMS crash course. We're well on our way through it. Same drill as last time. As you probably remember, we've explored kind of the big picture stuff about what is Bayesian inference and how we combine information from the prior and likelihood to get the posterior, which is what we actually care about. And we've covered all kinds of models so far, all kinds of generalized linear mixed models, different response distributions, different setups in terms of random effects. Then we've looked at these different ways to deal with space and time pattern in our data. What are we going to learn today? Well, it's a bit of a grab bag because, you know, not all data sets are created equal and all response variables are different. So today we're going to learn what to do with kind of weird distributions of response variables that don't fit the assumptions of some of the common statistical distributions we can use in GLMMs, where we actually have to make sort of little modifications to the distributions to get them to fit our data. And we're going to look at what to do if we have proportion data that's continuous but can only be between 0 and 1. We're going to look at what to do if we have count data, but it has a lot of zeros in it, as many count data sets have. And we're also going to look at what to do with continuous data and proportion data that includes zeros. There's various different models we can use. As we go, you're going to be introduced to the concept of a mixture distribution, and that is what we use to deal with these kind of weird distributions. And we're going to also look at how in a linear model, generalized linear model, not only Bayesian, but all generalized linear models, we can actually fit each different part of a statistical distribution. Each parameter of the distribution can have its own fixed and random effects, not just the mean. And yeah, as I say on the slide, all the stuff you're going to be learning today can be done without Bayes. It can be done with maximum likelihood, your classical statistics, but especially when you get to these weird and complicated distributions, Bayes is often the only feasible option, the only way that actually works in practice. And that's why I prefer to teach it that way. Although much of what we're learning today could be done without it. So let's talk about proportion data and beta distribution models to fit that kind of data. If you remember back to a couple of weeks ago, we had data that was like a proportion of seeds that germinated in each tray in some kind of experiment. And instead of fitting our model to those proportions, we actually fit a model to the binary data, number of seeds that germinated out of the total. Because we actually knew the actual counts of seeds. But what if you don't have access to those actual count data, or you truly do have a continuous proportion and you don't actually have any counts? You've got data that's between 0 and 1, but it's continuous. Well, you could try to fit a just classic linear model with normally distributed error, but that won't necessarily work because proportions have strict bounds between 0 and 1. Having that symmetrical bell curve error isn't good for that because bell curve doesn't have an upper and lower bound. It can go however far in positive and negative directions. So normally distributed error is often going to give you bad predictions if you fit it to proportion data. It can work okay if you've got proportion data that's kind of like not too close to 0 or 1. It's all between 0.25 and 0.75-ish, but in reality, in ag and other biological sciences, not a lot of proportion data really looks like that. So it often won't work to assume normal error on proportion data. So what should we do instead? Well, we've seen binomial so far, but that's not going to work in our case because binomial, like in the top figure, only has two discrete values, 0 and 1, that it can take. It doesn't handle this continuous variable like we have a continuous proportion. We've seen gamma. We used gamma to deal with data that was positive before in I think lesson 2. But that's not going to work for our 0 and 1 proportion data because gamma, yes, it has a lower bound at 0, but it doesn't have an upper bound. Like in the bottom figure, the gamma distribution goes to positive infinity. So what we need is a distribution that is only restricted to be between 0 and 1, and that can be very flexible in its shape within that boundary. So what distribution do we use? We use a beta distribution. It's a very cool mathematical function because it can take all these different colored lines here are different beta distributions with different parameters. Depending on what parameter you put, it can look like a bell curve. It can look like a skewed curve, like this purple line. It can even look like a horseshoe. This black line is also a beta distribution, but it always is constrained to be between the values of 0 and 1. So it can pretty much take almost any shape you need it to, but it's stuck between 0 and 1. That's exactly what we need for our proportion data, right? What the beta distribution has two parameters. If you look on Wikipedia article for beta distribution, it calls them alpha and beta, which is kind of confusing because the distribution itself is called beta. The R within R, there's the d beta function uses that same equation, but it calls them shape 1 and shape 2 for whatever reason. Well, BRMS changes around the parameterization. It's not the same as you would see on Wikipedia. It's rewritten to be in terms of two parameters, mu and phi, which is the mean and the precision parameter of the beta distribution. And this is actually very common for BRMS. The developer of BRMS and Stan wrote all the distribution equations so that if they have two parameters, like say the normal distribution, right? It's got mu and sigma, mean and standard deviation. The first parameter is more or less the mean, the expected value of the distribution, and the second parameter is the precision or the spread of the distribution. So or dispersion, some people call it. So like you can, and all the distributions are written that way. So we can say, okay, let's separate out effects on the mean from effects on the spread, basically. And the cool thing in BRMS, you can put fixed, random, whatever effects you want on any parameter. We can have fixed effects on the mean, random effects on the mean. We can have fixed effects on the dispersion or spread parameter and random effects on it as well. And we're going to look at that in the case of the beta distribution. Give me a thumbs up if that makes sense to you. If not, feel free to ask a question. Okay, I see people are nodding or whatever. So you understand we have a mean and a spread parameter, and we can put effects on both of them. What are we going to use to illustrate our example data set? Well, this is also from the very useful AgriDat package. Snyder's et al. Published this data, and basically they wanted to look at factors influencing how much leaf area on winter wheat was infected by a fungus Fusarium, and they wanted to know which of 4 different Fusarium strains were most virulent in terms of how much they would infect, and across 17 different wheat genotypes, and they did this in 3 different years. And right, because you are visually estimating the percentage of leaf area that's infected on a particular plant, that's a continuous variable between 0 and 1. There's nothing they're counting. They're just estimating, okay, this leaf is about 50% infected, this leaf is only 20% infected, and so forth. So it really is a continuous proportion. Okay, so let's go back into our studio session and get ready to work on lesson 4. So I would recommend going to session and restart R to clear away the variables and everything, just to make sure we're starting from a clean slate. Okay. And then in my Files tab, I'm going to go to the Worksheets folder, and I'm going to open Lesson 4 worksheet. Okay. And then I will highlight the first up to line 30. That will load all the packages and set all the options, and hit Control Enter. Okay. Now I'm going to load the Snyder's Fusarium example dataset using the data function, because it's preloaded in the package. Then we're going to do a little bit of processing to the data. Oh, here, let's just look at the data real quick. I'll type in my console head, parentheses, Snyder's. If I start typing the name, I can hit tab to complete and hit enter. So yeah, there's 4 columns. There's this genotype column. There's 17 different values. Strain, there's 4 different values of the genotype of the fungus, and there's different years that they took the data. Y is the percent infected. So you can see it is actually ranging from 1 to 100. It's a percent. First off, we want these first 3 columns to be factor variables. That works good for our plays well with our modeling syntax. So we're going to say across, and then which variables are we going to convert to factor? We're going to use C to define a list of column names to convert to factor, which is going to be GEN for genotype, strain, and year. And because beta distribution requires values between 0 and 1, not 0 and 100, I'm going to make a new column called P and create that by dividing Y by 100. P equals Y divided by 100. So we're going to convert the first three columns to factors, make a new column between 0 and 1. Okay, and now I am going to plot the data as a histogram, and we're going to use the variable P that we just created is going to be what we're making the frequency histogram of. And just for fun, I colored it in by strain, and then also separated the strains onto different panels by putting facet wrap strain. And when you do geom histogram and ggplot, by default it gives you 30 bins, but there's not that many rows in this data. It only has about 200 rows, so 15 bins per strain is plenty. So we can hit Control Enter to look at what does the data distribution look like. So you can see, just as I told you, we have proportions ranging between 0 and 1, and they kind of look somewhat like a beta distribution would be a good fit because they're kind of these skewed humps, skewed toward the 0 side. Except for this one is a little bit more spread out. F39 has some very highly infected individuals. Other than that, there's kind of a lot of low ones. And can we use Y? Oh, I have a question. Can we use Y percentage as a response variable with gamma distribution? You might as well, if it's a percentage, if the percentage goes above 100%, then you cannot use beta distribution anymore. You would need to use, yes, gamma distribution would be a good one to use. But if it's between 0 and 100%, you might as well divide by 100 and use the beta distribution, because dividing by a constant does not change the shape of the distribution at all. It's just changing the units. It's fine to convert units. It doesn't change anything about the data. So I would say if it's percentage between 0 and 1, go ahead and, or between 0 and 100, go ahead and convert to a proportion, and then you can do the beta regression model on that. Thanks for the question. The next thing I wanted to do before we start model fitting is not only to plot the distribution of the data, but also to look at how the levels of infection differ by strain and by year. So we're going to put year on the x-axis in this next plot. And we're going to put P, the proportion of leaf that was infected, on the y-axis. We're going to make a separate color for each strain. And this is what they call a strip plot, because we're going to plot all the data as individual points that are jittered, and we're going to add a median summary line in front of each one, which I think is better than a box plot for small data sets, because if you have relatively few data points, you might as well plot them all instead of summarizing them with a box plot. So then people can actually see all the data. That's what I typically recommend if you have only less than a couple hundred data points. And I've done some tweaking to jitter the points and also move them so that the different strains are separated. geom_point for the points. And I've jittered them to move them randomly, and then dodge to separate out the strains. And we're going to plot the median as a little line in front of each one. Control Enter to run that. So fun equals median. We're going to plot the median summary. So here we can see these are the 3 years, and within each year we have 4 strains. These are all the data points, and then the lines are the median for each strain. We can see that in some years there's kind of overall relatively low infection, and the medians for each strain is similar, like 1988. But 1987 and 1986, 3 of the strains have low infection, but this one strain in green, F39, had a lot of high values in those first 2 years. So it's kind of seems like there's a year by strain interaction, right, where the difference between the strains is different in some years, and there's little difference in others. But it seems mostly to be driven by this F39. So let's see what formal statistical evidence we can marshal to see what the difference is between F39 and these other strains. So what we're going to do is put together our beta generalized linear mix model. And we're going to use the BRM function as we always do. And P, the proportion infected, is our response variable. And the fixed effects is strain asterisk year. So we have strain, year, and the interaction are our fixed effects. And I'm going to include in this model a random effect of genotype. So maybe we care about comparing the different wheat genotypes, but at least for this example model, I'm going to say we don't really want to consider the difference in means of genotypes. We just want to account for genotype variability in the model so that we can test whether these fusarium strains have different virulence. Again, that may not be your research question, in which case you might want to put wheat genotype as a fixed effect. But for this model, we're really trying to compare the fusarium strains over time. And that's what we're interested in. That's why those are the fixed effects. So I'm going to include a random intercept for genotype and also a random slope with respect to year. So maybe not only are the genotypes different, maybe they differ between years in different ways. So that's what that random slope is. 1 plus year, a random intercept for genotype and a random slope by year. And then on the right side of the random effect statement, we put GEN for genotype. Okay, the response family is beta. It's got a capital B. That's how it's written in statistical notation. So we use the capital B beta for this one. It has a link function, just like we've seen in other generalized linear mixed models, that translates between the scale that, right, because the response can only be between 0 and 1, but a linear combination of fixed and random effects could take any value, positive or negative, right? We're just adding up fixed and random effects. There's no reason why that has to be between 0 and 1. So we need this link function to translate between our scale that can be any number and our response scale, which has to be between 0 and 1. And we're going to use the logit, the log odds link function. If you remember back to 2 weeks ago, we talked about the logit function, which can basically take a value between 0 and 1 and map it to a scale that can take any number. And we have a link function on our phi, on our dispersion parameter as well. These are all defaults that you wouldn't necessarily have to type these in if you're fitting a beta model, but I like to type them in explicitly so that you can see what's going on. The log link function is used for the dispersion parameter. That is the default that BRMS is using, but I just typed it out so you can see that. What prior are we going to use on the fixed effects? Well, again, as usual, we're going to not bias ourselves one way or another by saying we had a prior expectation of one genotype strain having a higher mean than another. So we want a normal with a mean of 0, right? Because we're not biasing it up or down. And I put a standard deviation of 5 on this prior. Again, it's not going to be too sensitive probably if I put 3 or 2 or 10, it would probably give very similar results. 5 is kind of a conservative number, but most likely other numbers in that order of magnitude would give us very similar results. And class equals B because B is the notation used for fixed effects. And then we're using kind of default or common defaults for the number of Markov chains and number of iterations. Let's hit Control Enter. Hopefully this won't start to compile again. Hopefully it will load the pre-fitted model object. Okay, so it's loaded the pre-fitted model object because it did not detect any change in the data or the model since I last fit this model. Can everyone give me a thumbs up if you're following me? If not, please ask a question. I'm here to help. Very good. Let's do a posterior predictive check. Again, that is a quick diagnostic plot where we look at the distribution of the actual data itself and the distribution of fitted values from different draws from the posterior distribution. If they have a similar shape, it's a good sign that our model is a reasonably good fit for the data. Control Enter to run that. And you can see decent, right? Here's the data, right? Overall, it's a skewed distribution that goes between 0 and 1. And the draws from the posterior, the blue lines, are also a skewed distribution that goes between 0 and 1, very similar in shape to the data. So we see nothing red flag about this. This looks good. But maybe we didn't capture every possible aspect of the data, because remember, the beta distribution has a mean parameter and it has a precision or spread parameter. We have only allowed the mean of that beta distribution to be different between the strains and the years. What if the variability, the spread, was also different between the strains and the years? Which, let me just go back to one of the plots I made. Yeah, like if you look, that may not be wrong, right? Because some of these strains have a much greater spread than others. Like F39 has kind of a wider spread. So maybe it's not just the mean that differs between them. It could also be the spread. What we can do is actually put a separate formula for that dispersion parameter with effects on the spread. So I'm making a new model. I'm going to call it fit with vary phi, because phi is the name of the precision parameter. Now I have a 2-line formula. So we actually have to use this function BF, which is BRMS formula. It creates multiple line formulas in the model. Most of the rest of the code is the same. The main model formula is the same, right? P, the proportion, is a function of the fixed effects, strain, year, and their interaction. And here's the random effects. We're also going to put the name of the dispersion parameter is phi. We're going to put fixed effects on it as well. Yes, Adam, do you have a question?
I don't know if you saw it or not. You have a question in the chat.
Oh, thank you. Our data is between 0 and 1, and we are using 5 standard deviations in the prior. Yes, thank you. I'm sorry I missed that. Well, the data is between 0 and 1, but the model is being fit on a log odds scale. So a standard deviation of 5, an effect size of 5 would represent 5 units on a log odds scale. You actually could get, like if you're looking at a difference between 0.001 and 0.999 on the log odds scale, that is pretty different. Like, yeah, let's actually the log odds function in R is Q logis. Let's say you had 0.99 for one of right, that's 4.5, and Q logis of 0.01 is negative 4.5. So you actually can get, because the model is on a log odds scale, even though the data on the response scales between 0 and 1, on the log odds scale, it can be very large positive and negative numbers. So yes, you could put 5 standard deviations as the prior is reasonable. Yeah, and it can't hurt to make the prior standard deviation high. All that means is that we are not constraining our expectation of what we think the posterior will be very much. So yeah, it can't hurt us too badly to make it a high number. Does that make sense? I'm sorry that I missed that question. And so what we're going to do for the second step for this model that's a little bit more complicated, we can see if we can get a slightly better model fit by allowing not only the mean, but also the spread to vary by strain and by year. So I'm going to put fixed effects of strain asterisk year on our precision parameter, dispersion parameter, sorry. And yeah, you could also put random effects and say, well, maybe this dispersion parameter could even vary by genotype as well. But honestly, with a small data set like this, I would say that is probably overkill, and it's likely to not to have a hard time converging because it'd be sort of overfit to the data. So we're just going to see, maybe you could try that, and I encourage you to try stuff like that out as an exercise. But in my opinion, it's typically enough to just put the fixed effects on this dispersion parameter and not put a whole nother set of random effects on that parameter as well. We have the same response family, the same data set. We have priors on the fixed effects on the mean, but we can now put a separate set of priors on the fixed effects that are on this dispersion parameter. And the way you would specify that is by adding this argument d par for distributional parameter equals phi. So which parameters fixed effects are we setting a prior on? And then that's what this is telling us. And then we can put in any prior distribution we want here. Again, we're going to use a normal. I've kind of mostly used normal with mean of 0 for most priors on fixed effects because it is relatively uninformed. And I don't remember my reasoning, but I put 2 as the standard deviation for this prior, probably because I don't expect to see huge differences in this precision parameter, in this dispersion parameter between the strains and between the years. So we'll just set it to a modestly low number to constrain the model and help it to converge, but still leaving the possibility open of that there is a difference in that. Yeah, and that's the model. And now we can run that. Because it didn't change, it loads the pre-fitted model object. We can do a posterior predictive check on this model. I'm going to hit Control-Enter to run this code and see if it looks any better or any different than the previous one. So it doesn't really look that different, again, because the other one already was quite close to the data. You can't really get much better. So this is very similar. Let's go ahead and do our leave one out cross-validation as we have done in previous cases to get a formal test of which of these models is a better fit to the data, given the number of parameters, right? It will account for the additional complexity and the extra parameters that we added when we said, does the dispersion parameter also vary by strain and year? If adding that extra complexity doesn't give us any additional benefit, the cross-validation will reflect that. But if it was worth it to add those extra parameters, our cross-validation will tell us that as well. And just like we did last time, add underscore criterion is the function we use to compute the information criterion for each model. We're using the loop. There are other information criteria you can use. The leave one out cross-validation, I think, has been shown to perform well for these type of models. So that's what I usually use. I'm going to hit Control-Enter. This is for the first model where we just allowed the mean to be different. And this is for our second model where we also allowed the dispersion parameter to be different. And then on line 91, I can do lu underscore compare to look at which one has a better fit to the data. Yeah, and so here, going back to your question, Brad, the one that adds an additional complexity is the best model, but the difference between the two models is only 10 in information criterion units, and the standard deviation is also about 10. So to me, that is kind of like a wash. I would say the two models are very similar. The difference, the point estimate of difference between them is about the same as the uncertainty in that difference. So I would say, well, and again, there's like, I should have included this, the formula to translate that into something like a p-value, which can be kind of informative. But this is, to me, not adequate evidence to say it's worth it to add that extra complexity. So we can just stick with, and again, when we looked at these model fit plots, they look kind of the same anyway. So it's unlikely that adding that complexity is really going to change our inference that much. So what do we do next? Well, the same thing we do for all our models, which is use them to make predictions and make inference about the different effects that we care about. So we're going to use our old friend EM means for that. And I'm going to look at the this tilde showing what is the form, which groups are we going to be estimating the means. Strain, Type year means give me the mean of each strain within each year, each combination of strain and year. And type equals response means I don't want those as log odds. I want them as the actual probabilities because that gives us the proportions that are interpretable. I'll hit Control Enter to run that line. And since I put parentheses around the whole line, it will also print it to the console at the same time. So yeah, these are just, you see that in 1986, we get predictions that are quite low for some of the strains and higher for strain 39. Again, that was a strain that had a higher observations in that year, which makes sense. And similar for the other years. How do we say, okay, is there statistical evidence that like this 0.24 is higher than this 0.59? We would have to take the contrasts, the pairwise contrast between each strain within each year to look at that. So we're going to use the contrast function to do that. Now, I had to add an extra step in here because the beta distribution is fit on the log odds scale, which means it will give you the contrast in the form of odds ratios. But to me, odds ratio does not make sense when we're comparing. We're not comparing two probabilities, we're comparing two proportions. We don't really want the odds ratio between the proportions. We just want the ratio of the two proportions. Like 0.24 is about 5 times higher infection than 0.05 or whatever, right? So we just want the ratio. To tell EM means to give us log scale comparisons, which is ratios instead of odds ratios, we have to use this regrid step and say regrid the EM means transform equals log. That will kind of coerce it to be on the log scale instead of the log odds scale, which makes more sense when you're comparing proportions. And the last argument is pairwise, because we want all pairwise comparisons between strains within each year. Then we'll run this code. And here, yep, so these are the contrasts. These are the ratios between each pair of strains. And so like this one, the 348 divided by 39 in 1986, that was the one that the 39 has about 4 times higher infection proportion than the other one. So that's why the ratio is about 1/4. And there's our 95% credible interval, showing that we are 95% sure, given our data, that the true value of that difference is between this pretty narrow range of 0.2 to 0.3. And if you want to do null hypothesis testing on that, again, this should be repetitive for you because we do it. We've done it multiple times already in this course. We can get first we can get the credible intervals on those means using the describe underscore posterior function. And we can get any credible interval percentiles that we want. And as I've said before, I like to do the 0.66 and the 0.95 because it's like 1 and 2 standard deviations away from the mean. Equal tailed interval. And we're not going to do hypothesis testing on the means themselves. We're going to do hypothesis tests on the difference between the means. So just for the credible interval of the means, I would put test equals null to not do a p-value there. Hit Control Enter. And then we get a nice table of the posterior estimates and their credible intervals. We can do the same for the contrasts. We get a credible interval on those ratios. And if you want, you can compare different forms of Bayesian p-value, like probability of direction PD and p underscore map for the maximum a posteriori p-value. And because we're testing the null hypothesis that the ratio is equal to 1, that there's no difference between the strains within a given year, we have to put null equals 1. A ratio of 1 means they're the same. So we specify null equals 1. Run that line, And there we have, for some strains, there's strong evidence of a difference given the year, especially the strain 39 is shown to be, we have strong evidence that it has higher infection than the other strains in that year. Versus in 1988, the differences, the ratios are closer to 1. They're 0.6 or 1.2. Some of them are still showing evidence for a difference, but the magnitude is smaller of that difference. Yeah, so if you go to the slides, I won't necessarily bore you with it, but you can see what I suggest for writing this up in terms of the methods and results section in a manuscript. And I've also included code here to make what I think is a nice looking figure that could go in a manuscript. So I'll just run that code to generate that. So yeah, and I've intentionally included the data and model predictions on the same plot, which I think is nice because it illustrates, here are the model predictions with their credible intervals, but also here's the data. So it's kind of a sanity check that the model is actually has some connection to reality, right? The model is not real. It's only a tool we use to learn about the data. So I think it's often nice to include not just those nice means with error bars that look all clean, but also the data, because it shows some of the actual variability that's in the real observations. Not to mention, it can help confirm that the model is reasonably good because, we don't see some weird discrepancy between the model and the data. So I've put them both on the plot with some nicer formatting for like you would put in a manuscript. And here is a table. If you run this code, you can generate a table of these contrasts and the Bayesian style p-values and the upper and lower bounds of a credible interval for all of them, which may be something you're interested in presenting if you were specifically really getting into how the relative difference in these strains' virulence change from year to year. Okay, what questions do folks have about that? Give me a thumbs up if you're good. It's too bad Teams does not have a thumbs down or some kind of negative reaction because that you can put in front of your face, because then I could see if people were, yeah, there you go, a shocked face. Perfect. I know Zoom has like the green check and the red X, which is kind of nice, but anyway. Well, I have a bunch of little kind of like small examples in this lesson. So I'm just going to go ahead and do, since we just took a break a few minutes ago, I'll do another one. Let's just take a short break in case people need to stretch or something. Let's take another 5-minute break and we'll get back at 3:38 Eastern time. There's no point in like trying to drive this thing too fast. We have time. So let's take a 5-minute break. See you in 5 minutes. If anyone has any questions now, feel free.
Hey, Quentin, I have a question.
Yeah, go ahead.
Yeah, just philosophically speaking, when you're showing these beta models, what
Right.
it seems to me, and because I deal with this a lot with my data, is I think, like proportionality data. Is there ever a time where you really wouldn't want to use this if you knew how to use it? It seems like it's more reliable than any other method that you've covered for that kind of data set based on what
Yeah.
I'm understanding.
I mean, to be honest, yeah, it is beta is like the only statistical distribution that has the properties that like actually work for this kind of data. The reason not to would be right, if you have a small sample size and are having trouble getting that model to converge because of, like the issues we've been dealing with the data set we've been working with, then you can just like kind of like bail and say, okay, I'm just going to like log transform and fit a normal error model to the thing. But if you have Bayesian tools, then you don't ever have to do that because you can always get the answer, if you use priors. But like if you really don't want to use priors, then and you can't get the model to converge, then you will have to make some kind of compromise like that. Yeah.
Okay.
Mainly the reason would be, yes, if there's numerical issues with the model converging. Other than that, yeah, why wouldn't you use it? I mean, there's really no good reason not to.
Okay. But yeah, I specifically meant more like these kind of beta models are the only ones you would use, not anything you've covered before, right? Then,
Yeah, right. Yes. Yes, if you're doing a GLMM on proportion data, I think you would want to use the beta model. Yep.
okay.
It's because it's the only one that is bounded on two sides.
All right. Thank you.
Sure. Okay, so we'll start up again in just a moment. I will share my screen again. So yeah, the next thing we're going to talk about, yeah, this lesson is a bit of a grab bag because it's kind of just hitting up all kinds of different distributions that are ones that I have work with all the time in ag science and think that you also might find relevant, but there's just a little unique types of models for specific situations. Another one that comes up a lot in the kind of work that we do is counting things. And when you're counting things, sometimes there's no things to count and you have a zero. We learned a couple weeks ago about how to fit a Poisson model to count data. But Poisson distribution has a very specific shape, and sometimes there's more zeros in the data than a Poisson distribution can handle. What do you do in that case? So I'm going to give a few background slides before we get into the example. So um, as we've talked about before, we've talked about gamma and beta distributions, right? They support positive values. And so far in the examples I've purposefully chosen when we did gamma and beta, I purposefully chose examples that did not contain any zeros, right? Because gamma and beta only supports values greater than zero, not zero. But obviously, you don't always have the luxury of getting to choose an example data set. You have to work with the data you have. Lots of proportion data has zeros. Lots of count data has zeros. Lots of continuous non-negative data like concentrations has zeros, sometimes a lot of zeros. So what do you do if you have all those zeros? Well, like I said, I've said several times before, there's an old school way, make the data fit the model, which works okay in many cases, but it's not, I think, the most scientific way to do things, right? Let's just get rid of the zeros by adding a little bit to all our numbers and then possibly transforming the data as well. Like sometimes there's legitimate scientific reasons to do this. For example, if you have concentration data, but there's a limit of detection, like your instrument is not sensitive enough to detect concentrations below a certain threshold. Well, there's no such thing as true zeros. There's always going to be at least one or two atoms of your substance that you're looking at the concentration. So maybe they're not really zeros. So some people use some like half of the limit of detection and replace all the zeros with that. But other times, there's not really a good biological reason to replace the zeros with a positive number. And so people just pull a number out of any place, like do we do the log of x plus 1? You know, because right, you can't take the log of 0. So do we add 1? Do we add 0.01? And so forth, right? And depending on what fudge factor you choose, it's going to put bias into your results, not to mention the bias introduced when transforming and back transforming, like I've already talked about. The other old school way of dealing with it is just deleting the rows of your data set that are 0 and then fitting the model to all the non-zero values, which is honestly, I still occasionally do that. If you have like thousands of rows and maybe 2 or 3 zeros, it's not going to make a big difference, and it's overcomplicating things to make a special model to deal with the zeros. So sometimes it can be acceptable, but you'd better have a good reason to do that, right? To just delete rows from the data because they're inconvenient. What do we do? What's the best way to deal with these zeros? Well, we're going to resort to what is called a mixture distribution I mentioned earlier. A mixture distribution is any statistical distribution that is made up of 2 or more distributions kind of squashed together, stacked on top of each other, and then renormalized by taking basically weighting a sum of their density functions so that the area under the curve is 1 again, right? Like if you take 2 normal bell curves, they have an area of 1 under each one. If you mix them together, then you have to just renormalize it so the area under that resulting mixture is 1. You don't have to just mix 2 bell curves. You can mix distributions from different families, and it doesn't have to be equal. You could mix it at different proportions. Here's a couple examples. This is what it looks like, a mixture distribution between 2 normal bell curves. One has a mean of 0 and a standard deviation of 1. One has a mean of 3 and a standard deviation of 1, and we're mixing them at equal proportions. It kind of looks like a double humped 2 bell curves stuck together, right? If we mixed it at an 80 to 20% proportions, obviously the one that mean at 0 is a lot bigger of a peak than the one that has a mean of 3 is just a little shoulder anymore. We could mix 2 gamma distributions with different parameters at a half and half rate. That's what that would look like. And we could even mix a normal and a gamma if we wanted to. Here's the normal and here's the gamma, and they're mixed at half and half. You could do whatever. What is the application of all that to our current situation where we're trying to deal with zeros? Well, we can use a mixture distribution for that because we can model the excess zeros and sometimes the ones, if you're talking about a beta distribution, that cannot be fit by our standard statistical distribution. And I always think this example from the book Statistical Rethinking, which I highly recommend. I've mentioned it before. It's a very good Bayesian tutorial by Richard McElreath. He gives this good example that maybe help you understand. So back in the Middle Ages, monks used to work all the time copying manuscripts, right? They had to take one book and copy it onto another piece of scroll. And that's all they did all the time, right? All they did was work on copying manuscripts, but manuscripts are very labor intensive to copy. It takes many days to copy one. So like an entire monastery full of monks might be working all day and only finish one manuscript. They might finish 2 or 3 on another day. Some days, since they take so long to copy, they might not finish any, even though they were working all day on it. Or it could be a day where they were taking a break and drinking beer and didn't copy at all. You always get a 0 finished manuscripts on those holidays, on those beer drinking days, right? But you still could get a 0 even if they were working, because they just might not have finished one that day, right? This is what the distribution of manuscripts finished per day would look like. You would, anytime that they finished at least one, it's a work day, right? Because they finished at least one manuscript. But the 0s, we don't know necessarily why it's a 0 that day. It could be because they were working but didn't happen to finish one, and it could be because they were drinking beer and weren't working, right? It's a mixture of the two. We can model that. We don't need to know for any particular 0 which one it came from, because all 0s look the same to us. We just have to assume that there's a greater proportion of 0s in the data than we would expect for a count distribution such as Poisson with a given mean. We can basically juice the Poisson distribution by adding more 0s than would be. Poisson distribution has only one parameter, the mean. And if you have a given mean, you have a certain proportion of 0s. We can say, well, we're doing a Poisson distribution, but adding in this extra bit for more 0s than we would expect. Poisson is a discrete distribution, right? It has only integer values, 0, 1, 2, 3. So there's sort of a finite probability of getting exactly 0 if you take a random draw from a Poisson distribution. The model we're going to do now is called zero-inflated, because we are inflating the probability of getting 0 from that distribution with an extra parameter. And this is a mixture distribution of a binary distribution of 0 and not 0 with our Poisson distribution. There's a slight variant of that is for continuous variables. Like if we had a gamma distribution, that's continuous, right? It does not just 0, 1, 2, 3 values. It can take any continuous number. Beta is the same between 0 and 1. You can't define the probability of a continuous variable at one exact. There's no probability of it being exactly 0. In fact, there's no probability of it being exactly anything. You can only define the probability over some interval using calculus, basically. So for that situation, we don't call it zero-inflated model anymore, but we can still model having excess 0s in that data set. We basically use a hurdle model. It's a similar concept, but the term you'll see in the literature for that is called hurdle model. We're still doing a mixture distribution. We're doing a distribution that has a sort of point mass at 0. That's the probability of the 0s. And then mixing that with our continuous distribution for the non-zero values. So we can fit a gamma or a beta that has extra 0s in it. Some of it being exactly 0. So we can do this for discrete count data. We can do it for continuous variables. And we can do it for proportions with a beta distribution. We can always deal with those 0s. Can everyone give me a thumbs up if you're following what I'm saying here? So yes, and this is not just academic, right? These are things that we see all the time in the data that we work with every day. So this is something that's kind of important to be able to deal with. And so zero-inflated Poisson is the first type of zero-inflated distribution we're going to do. It's like the monks drinking beer example we're going to do, but it's in a more ecological or agricultural context. You often have to count the abundance of some organism, right? Like insects on a crop or something. A lot of times you don't find any, so you get a 0 observation. there are Poisson and also sometimes negative binomial distribution. Those are two distributions that are discrete and work well with count data, but they don't necessarily have enough zeros in them to work with every data set. So they do have some probability of zero, but we need to add an extra parameter to inflate that probability. In other words, we're modeling it as having two kinds of zeros, some from the main distribution and some from that extra inflation component. Just like if you go back to the monks drinking beer, there's two kinds of zeros, right? Zeros from when they were working but didn't finish, and zeros from when they were kicking back and having beer. There's two kinds of zeros. And here's the example data set that we're going to use for the zero-inflated Poisson. It's a classic data set by Beal from 80 or 90 years ago, where they wanted to figure out how to control this caterpillar that's a big pest, the beet webworm, and they wanted to know different ways of killing this thing. So they I don't know what they did back in 1940, but they had spray pesticide and they had lead. And so they wanted to know if you could use spray or lead or both to control the beet webworm. So they did a 2x2 study where you have, spray, a control group, spray, lead, and both, a 2x2. And then they had a plot for each of those combinations in a block, and the whole thing was replicated 4 times in a randomized complete block design. And each plot had a bunch of plants in it, I guess, and on each plant they counted how many egg masses of the beet webworm did they find. They did that for 25 locations, I guess 25 plants within each plot. So it's a count. It is a discrete value, an integer, 0 or more is how many they found. So let's go into our RStudio, and we're going to start exploring this data set and then think about what kind of model we're going to fit to it. So let's go back to our RStudio server. We can just scroll down. Here on line 125, I'm going to hit Control-Enter to load Beal webworms into my workspace. And then I'm going to plot the distribution of webworms on each treatment combination. And x equals y because the name of the variable in the data set is y, but we want it on the x-axis for the histogram. We're going to do a histogram, and then we're going to do a facet grid. So we're going to have 2 panels, a 2x2 grid of histograms for each level of spray and lead. So we can do spray, S-P-R-A-Y, spray by lead. And I've added some code because in the data, they're just called Y and N, but I've labeled it as no spray and spray, no lead and lead, so you can see what we're dealing with. And there was anywhere from 1 to 9 egg mass in the on each plant that they counted. Oh, really? It should be 0 to 9 since we do have some zeros in the data set. So let's just change that to 0 to 9 and run that. Yes, so here are the distributions of counts. You can see that in all 4 of the treatment combinations, we have a reasonably large proportion of zeros, right? And there were as many as 9 egg masses on some of the plants, but most of them had 0 or a small number. So let's say we don't really know whether or not Poisson distribution can handle some zeros. We don't really know whether a zero inflation term is really needed in this case. So why don't we just go ahead and fit our Poisson generalized linear mixed model to the data and see how we do. So y, the count, is our response variable. Our fixed effects are spray treatment and lead treatment. and an asterisk for their interaction. And well, I should have showed you what the data looks like. Let me type in the console head parentheses field.webworms. You can see there's row and column coordinates, so there is a spatial component to this, but we're not going to worry about that for this example. Of course, you could do something like we did in the previous lesson, but we're not going to worry about that right now. There's the Y variable, that's the count. There's blocks, and treatment is like there's 4 levels of that for combination of spray and lead, but it's also split out into spray and lead, which each has 2 levels, no and yes. But you can see that we actually have a lot of, that's right, the row and column are coordinates within each plot. So those are like, each plot has a bunch of locations within it that plants were counted. So first of all, since this is an RCBD, we need a random intercept for block to account for any systematic variation between blocks. So we'll do 1 by block. And because each unique plot has 25 repeated measurements in it, we also need a random intercept for the plot because the measurements within each plot are not independent. And a plot is a combination of treatment and block. And so our treatment is identified by spray and lead combination within each block. So to identify our plots, we're going to say the combination of spray, lead, and block. We'll do a random intercept for each plot. Spray, lead, block. Now, of course, you could create in your data set a column for plot ID and then just put plot ID as the random intercept if that's makes more sense to you, but it would have the same effect. And we're going to use a Poisson response family with a log link function because that is suitable for count data. And as usual, I'm going to throw a normal prior with a mean of 0 and a standard deviation of 5. Again, any small whole number will do because this is on the log scale. A 5 log fold change is huge, so that's a very uninformative prior. And we're using kind of common defaults for the number of MC iterations and so forth. I'm going to hit Control-Enter to run this. So again, we have not accounted for zeros beyond what a Poisson distribution can normally handle in this model. And to see how well does the model fit the data, we can do a posterior predictive check plot. And because we don't have a continuous response, you can make the posterior predictive check plot for discrete responses by using a bar plot instead of one of those continuous density plots. So type equals quote bars is what you would use for that. Hit Control-Enter to look at the fit. Okay, so what do we see here? The bars are the observed data. So these are like the counts of zeros, ones, twos, and so forth that we got in our actual observations. And the dots with error bars are the fitted values over different draws from the posterior distribution. So you can see here that the number of zeros observed is higher than what the model is predicting back. And to compensate for that, the model is predicting more ones and a little bit more twos than we actually observe. So there is a discrepancy between the model predictions and the observations. The model is not letting there be enough zeros to accommodate the data. So what do? Let's make a model that fits the data a little bit better. And how do we do that? We're going to throw in an additional line in the BRMS formula, just like we did when we added more lines to the beta regression formula. We're going to use the BF function for a multi-line formula. The first line is exactly the same as before. We have our response variable Y, spray, lead, and the interaction or the fixed effects, and here are the random intercepts. Well, in BRMS notation or in syntax, whenever you add a zero inflation term to a model, the name of the parameter that governs how much zero inflation there is called ZI. So we have to make a second line in our formula for ZI. You have to use that name because it's not from our data. It's actually a name of a parameter in the model. And I'm just going to put fixed effects on ZI. I'm going to allow the proportion of zeros to vary by treatment, but I'm going to say it's constant across the blocks because again, you could add random effects on the zero inflation parameter, and I encourage you to try it as an exercise, but in most cases, it will be kind of overly complex and overfit the data. We don't really have enough data of each block to look at the difference in the proportion of zeros, but we do want to know if it's different by treatment as well. Spray asterisk lead to put fixed effects on our zero inflation component of this model. Now we cannot just use Poisson anymore as the response family. BRMS has a special built-in zero-inflated Poisson response distribution, which is zero_inflated. You can see that the tab completion starts, so you can see all the zero-inflated distribution it supports. Zero-inflated_Poisson. We're still using a log link for the main response, but the zero inflation also has a link _ZI. The default is to use a logit, log odds for this, because zero inflation is basically a probability, the probability of observing a zero. And we usually use log odds or logit link function to translate probabilities into a log odds scale that can take any number. And now, because we have priors, or because we have fixed effects on our zero inflation parameter, we also need to put priors on those fixed effects because there are new parameters introduced into our model. This class equals B is for the fixed effects on the mean, and then our dpar, distributional parameter, is ZI. What are the prior distribution on the fixed effects on the zero inflation parameter? And just to be conservative, I've put a norm with a mean of 0. We're not saying which treatment has higher proportion of zeros, and I've put a standard deviation of 5 to be very conservative. That will accommodate very large differences between the treatments. It's not putting really any information in there through that prior. And we're using common defaults for the number of Markov chains and iterations. And I'm going to hit Control-Enter to run that. Now, let's look at the posterior predictive check plot for this zero-inflated model. Let's see if we can improve on what we have before, where it was underestimating the zeros. Run that. So now that looks pretty good, right? Like our posterior predictive is right on top of our observations. It is not overestimating and it's not underestimating the zeros at all. And same goes for the 1s, 2s, and 3s. So this is a much better fit to the data. I've left it as an exercise to do the leave one out cross validation if you want to do that. I don't know if anyone's actually doing these homework exercises, but all the lessons have little exercises at the end, and so that would be one that you could do. But yes, it does. The cross validation supports adding the zero inflation parameter because it only added a little bit of complexity and increases the quality of the fit by a lot. So that's how you fit a zero inflated model. And I've also included, as usual, a bunch of code for how you would use that model to make predictions and then to do tests for the effects that we care about, just like we do for every other model that we've done in this lesson. And there is a bit of a wrinkle to this when it comes to the zero inflation model, because a zero inflation model has, yes, let me actually show the slides for this because it's nice to see it. There's sort of 3 different predictions you can make from a zero inflated model based on the parameters that it has. One is the conditional mean. That is the model's predicted value of how many eggs would be predicted for each treatment on average, conditional on the non-zero. So like if there's at least some eggs, what is the average number of eggs that are there? We could also use the model to predict the zero inflation probability for each treatment. What percent of the time on average would we expect there to be eggs versus no eggs given the treatments? But if you multiply that together, you'll get kind of the overall response, like the average number of eggs you expect to find, accounting for the number of eggs you see times the probability of there being at least some eggs. It's like the conditional mean times the probability of non-zero is the overall average number of eggs, which I think most times, I think scientists, at least in ARS, are actually interested in this overall mean. They want to know, they don't want to have to separate the zeros from the non-zeros and give two different predictions. They just want one overall prediction. So usually when I'm working with scientists on this kind of data, I will give them this overall mean and multiply stuff together. Now, luckily, EM means has syntax that allows you to get each of these three different kinds of predictions as estimated marginal means, and I'll show you how to do that. I'm going to do the conditional mean, the probability of zeros, and the overall response. EM means is the function every time, and we're going to do for each combination of spray and lead, the average number of the estimated marginal mean for eggs. And we're going to do, if you just do type equals response with no additional arguments, that's just going to give you the conditional mean. It's not going to account for the zero inflation part of it. So I'll hit Control Enter to look at that. So here, and this is back transformed from the log scale. So like if there's no pesticide at all of any kind of the control group, there's an average of 1.6 eggs per plant, and with a credible interval here, versus if you do both pesticides, you get a 0.5 eggs per plant with a credible interval between 0.3 and 0.7, 95% credible interval. If there's at least some eggs. This does not account for the probability of zeros. What is the difference in the proportion of zeros we would expect to observe between the two, between the four treatment combinations? We add an extra argument, d par equals zi, and that will give us the estimated marginal mean of the zero inflation probability for each of the treatment combinations. Control Enter there. And here we have it. So there's, this is like 17.6% expected proportion of zeros in the control group versus more in the double pesticide group, as we might expect if it's controlling the insect. How do we get the combination of the two? We have to use the argument EPRED, expected value of the posterior predictive, equals true, and this will give us the overall response, accounting for both components of the response. Control Enter for that. So now you can see it's 1.3 on average for the control group and 0.38 for the double pesticide group, which if you look back is a bit lower than the conditional mean, which makes sense because this is accounting for some zeros, so it's going to make the number go down a little bit. And so this is kind of the difference overall. Again, here are some ways that you could then do hypothesis tests on that and how you might present it in a manuscript in terms of a figure. And I've got in the text version of the lesson in the slides, text description of what you'd put in the text of the manuscript. Again, to do the contrast properly between the treatments, we have to do this regrid step again because once you kind of combine the conditional mean and the zero inflation probability, EM means loses track of the fact that a log link function was used. So we have to re-specify that a log link function was used to get the correct contrasts of ratios of counts between the treatments. So we have to say regrid transform equals log, and then we'll pipe that into a contrast function, getting the pairwise contrast. And if let's say we wanted within each lead treatment, is there an effect of spray? We would say bi equals lead. And that will give us within each lead treatment the difference between the two spray means formulated as a ratio. Control enter there. So yes, the ratio of number of eggs in unsprayed versus sprayed plots, if there's no lead, is about 2.7. So there's more eggs if you do not spray, which makes sense. And a kind of a similar ratio if there is lead, right? And what if you wanted to know if you spray versus not spray, does applying lead have an effect or not? We're going to do the same exact syntax as last time, except instead of bi equals lead, we're going to put bi equals spray. Control enter there. Here we have, if you do not spray, no lead versus yes lead, there's a ratio, there's the ratio of, so the effect of lead seems to be smaller than the effect of spraying because it's a ratio of 1.6 versus a ratio of about 3. Now, if we do spray, the difference between no lead and yes lead is smaller. The ratio is only 1.2, and the credible interval is actually 1 is contained within our 95% credible interval. So there's less evidence for an effect of lead on the caterpillar numbers if you spray. So maybe if you've already sprayed, adding that extra lead treatment is not doing as much since there's not as many. The spray has already dealt with the caterpillars for the most part. And we can, as I've shown you in previous examples, we can also get a Bayesian style p-value for all these contrasts as well. Just like in last time, we used describe underscore posterior, get that p-value, and we can get a equal tail 95% credible interval on it, on the contrast. And we can use the p underscore map and the PD to get different ones. It's sort of up to you which one you want to present in a manuscript, and you can look at the literature for justification on either one. I've kind of used both in different contexts, and we're going to say the null is 1, right? Because the null hypothesis we're testing with these Bayesian p-values is that the ratio between the treatments is 1. So we're going to do that for our spray contrasts. You can see we have very strong evidence of a difference, an effect of spraying on caterpillar control, which makes sense. And what about lead? Here, this kind of matches what we saw before. If you do not spray, there is strong evidence for an effect of lead on the ratio on the difference between the caterpillar numbers in no lead versus yes lead. But if you do spray, we have little evidence, pretty much no evidence that there is a difference between them. So again, if you spray, you don't really need to also do the lead as well. And again, I put in the slides text for methods and results, and here's a figure which could be used to show everything we've done so far. Now, this is, you may not agree that this is a good figure. I kind of like it because it shows the data as a histogram for each treatment combination and the mean with a credible interval around it. Yeah, your mileage may vary if you like that or not, but it kind of shows you the raw data and also the distribution or the uncertainty in our estimate of the mean. So like here's the control group. It's got a lot of caterpillars. And here, if you spray, whether or not you use lead, there's not very many caterpillars if you spray, regardless of lead. So that's kind of the take-home message there. So that's the end of that example. Yeah, why don't we take another? I need to take a break for a second. So why don't we take another 5-minute break and come back at 4:19, and I think we'll we'll have time to do another fun example before we adjourn for the day. So I will be right back, and then I'll answer questions when I get back. Or does anyone have any questions now, actually, before we take a break? Excellent. So I will be back, and we'll start again at 4:19. Thank you for your patience.
Hey, Quentin, I had a question about the conditional mean.
Yes, go for it.
Okay, so I noticed in the EM means output that some of
Yep.
those were below 1, which is
Yeah,
only
that's
possible if
just
there are
the
some zeros, right?
yes, the conditional mean. Yes, so Poisson has some zeros. It is not yes, a Poisson distribution has some zeros in it. It just because it only has one parameter, it is not flexible enough to have that many zeros with a given mean. So basically, like, our distributions are not shaped exactly like a Poisson distribution should be. They're kind of like heavier on the zero end. So that's why we add in the zero inflation parameter.
So it's not conditional on it not being a 0. It's like
Uh,
it's the mean conditional on it not being a 0 because of the
It's
weird
conditional on
breakdown.
it not being a zero inflation zero. That's right. Yes.
Okay.
So there's two kinds of zeros in the model. There's there's some of them are from the Poisson distribution intrinsically, and some are from the yes, that's right. I'm sorry. That was you are correct, and I said it in kind of a sloppy way or whatever. Yeah, like if you look at uh the posterior predictive check plot that's on the screen there, you can see This is the one from the model that didn't have zero inflation. It still predicts a very large proportion of zeros. So there's still plenty of zeros. It's just not quite the perfect fit. Yep.
Cool. Thank you.
Sure. And yeah, also there's also the negative binomial distribution, which a lot of people use, which actually can be used to fit count data with a lot of zeros, even without doing the zero-inflated Poisson. The negative binomial can actually accommodate more zeros than a Poisson. So some people use, and you can even do a zero-inflated negative binomial if you really have a lot of zeros. It just depends. It depends on the shape of the distribution of the data, which one is best. Cool. So yes, let's go ahead and do another example because you're here and why not? And this is stuff, that ag scientists deal with. So let's do one more example. Since these are distributions that we encounter and we have to work with. Now, what if so far we've done proportion data today, we've done count data, so discrete integers that have zeros, but what if you have continuous variable that's positive but also has zeros, like a concentration? What do you do then? So we're going to talk about fitting a hurdle log normal model. I'll give you a bit of background on that. Yeah, in natural science, it's very common to have data that has zeros but is non-negative continuous variable, like concentration, right? It's not possible to have a negative concentration, but your instrument cannot detect a concentration below a certain threshold, then you get a zero. For all practical purposes, it is zero. We've talked about using distribution like gamma for fitting positive continuous data. There's also log normal, which also works well for positive continuous data, but those distributions do not encompass zero. They do not accommodate values of exactly zero. In fact, they aren't discrete distributions, so they don't have any probability of being exactly any one specific value. They're continuous. So what do we do if we have zeros in a case like that? Yeah, just like I've always been saying, there's the old school way, and then there's kind of the new and better way. Back in the day, people, and still today, people would just add a little bit of a fudge factor and take the log and fit a regular linear model to the data. Or they might add a fudge factor and fit the gamma model, like get rid of the zeros, but still do the gamma model. But both ways kind of introduce bias and is kind of torturing the data to fit the model. So why don't we, as usual, it's better to get a model that fits the data, right? I think so, at least. So the hurdle model, we've already talked about it a little. It's very similar to a zero-inflated model, but right, just like we were just talking about with Grant, in the Poisson model, we did have a pre-existing probability of zero before, and then we're just inflating it. But in this model, we don't, in a gamma distribution or a log normal, we don't have any zeros intrinsically. So we actually have to add a probability of zero to a distribution that ordinarily can't have any zeros. It's called hurdle because it's sort of like you have to reach the hurdle of to become a non-zero value. You have to be higher than that probability of non-zero to have a non-zero value. Anyway, they just call it hurdle model in the literature, so that's what we're going to call it. And it's called that in BRMS as well. So this example I didn't actually get from the AgriDat package. I actually got it from an ARS scientist that I work with, so it should be very relevant. Hopefully people here that are, I've always used agronomic data so far, so this also will be good for anyone who does immunology and is not always looking at yield and stuff, because lots of people in ARS also work on this. This data set is from an immunoassay, and we basically are trying to develop a new vaccine for chickens and see how well it does. So basically the way the immunoassay works is you take the expectation is that if you vaccinate a chicken, it will be stimulated to produce a lot of antibodies in its bloodstream that will bind to the antigens on the microorganism that we are trying to immunize it against. So basically, if we have an unvaccinated bird and a vaccinated bird, the vaccinated bird should have more antibodies in its bloodstream that will bind to that protein. So the way the immunoassay works is we take some blood from a vaccinated and an unvaccinated bird. We mix the blood with a chemically labeled antigen that kind of glows when it attaches to an antibody. And then we can look at how much chemiluminescence, how much light is coming off of the chicken blood, and the more light comes off, the more immune response it has. I mean, it sounds like so elegant and simple, but the person who invented that, or the people that invented that, got the Nobel Prize. So, you know, once you see it, it seems obvious, but it was very revolutionary when these immunoassays were first developed. That's what they did on some chickens. And we're looking at, basically, this is kind of a technical study looking at what parts of the bacterium cell, what antigens the vaccine was most effective at binding to, basically. So it's like, is it the flagellum or the cell wall or whatever of the bacterium? So basically, we took 10 vaccinated and 10 unvaccinated birds, and blood from each bird was exposed. An aliquot of blood from each bird was exposed to 4 different antigens, 4 different proteins isolated from a bacterium. And it's a split plot design in the sense that we drew blood from a bird, split it into 4 aliquots. I shouldn't say we, I didn't do this. I just helped analyze the data. They did the same bird, which each bird only has 1 vaccination treatment. It's either vaccinated or it's not, but its blood was split up and exposed to 4 different antigens. The real study was more complicated than that, but this is just part of it. And the thing is that if a chicken is not vaccinated, its concentration of antibodies is so low that the luminescence produced by that sample is undetectable, and we get a 0. So there's zeros in the data set. And because this data is not in the Agridat package, we have to read it from an external CSV file. So let's go, and if you're not on if you're on SciNet, it's in a folder called data. If you're not on SciNet, you will have to read it from this place where I've posted it on a URL. So since I'm on SciNet, I can just run this line 190 to load the immunoassay data set into my workspace. And let's plot the data to see what we're dealing with. Antigen is the X variable. That's so each chicken blood was exposed to 4 different antigens. The Y variable in the data is called chemiluminescence. If that's too hard to spell, you can look at the immunoassay data set in your console, and it will show you the names of all the columns there. Right. So each chicken has an ID. It's either non-vaccinated or vaccinated. These are the names of the antigens. There's also antibodies, but we're ignoring that for the purpose of this example. And there's the variable. So you can see some are 0 and some are a large positive number, which is basically continuous. I think it's rounded, but it's a continuous variable. And we're going to color in the data points by the vaccination status of each chicken. And we're going to group by vaccination so that we can see the vaccinated versus unvaccinated for each antigen as a separate set of points. They're going to be moved apart from each other. Hit Control-Enter to run that. So here's our results. So we can see that, or our data. You can see that the orange is kind of like the baseline immune immunity of a chicken that doesn't have the vaccine. You can see that for some of these antigens, there is a non-zero baseline. For others, there's basically no immune response whatsoever in the unvaccinated chickens. But in the vaccinated chickens, there's a decent amount of response in most. But even in the vaccinated, there's still a few chickens that didn't respond well to the vaccine and still are producing no antibodies. They have zeros there too. That's what I did. So there's zeros in kind of a lot of the treatment combinations. In fact, some of them are all zeros. Every single one of these numbers here for this antigen for non-vaccinated birds is 0. I mentioned that there are 2 different continuous distributions. We might want to fit gamma and log normal. And to decide which one is a better fit, instead of fitting the full Bayesian model to each one, what I wanted to do here was just do a quick maximum likelihood fit of gamma and log normal to the data and just graphically look at which one is a better fit so we can decide which one to go with. So what I wanted to do was just basically take a subset of the values in this data set that are greater than 0 to look at what is the distribution of the non-zero values in the data set. So I'm taking the subset that's greater than 0. So we're only taking the subset of the chemiluminescence values that are greater than 0 here. And then I'm going to use the fit dist function from a package I loaded earlier to fit a statistical distribution to a set of data. And we're going to do gamma and log normal. And the name of the distribution is gamma. And for log normal, it's L norm in this particular package. And then I've made a plot to graphically look at the fit compared to the data. So I'm going to highlight all this and run it. And so here's the orange line is the gamma distribution, and the blue line is the log normal. And it's a pretty small data set, so the fit's not going to be perfect because there's a lot of noise in this small data set. But to me, just graphically, it looks like the log normal, the blue line, is just a tiny bit better of a fit. It's kind of got the mode of it is closer to what we actually observe in the data versus this gamma has a little bit lower of a peak and a little bit more spread. So we're saying, okay, maybe log normal is, let's just go with log normal. Again, in the exercise, you can fit the gamma model as homework if you want to really compare them more rigorously, but we're just going to do log normal because based on this eyeballing here, we're saying it's a better fit. So how do we fit the hurdle model to the data set? Again, I'm just going to go straight to the hurdle model this time. BF, because we have a 2-line formula, one for the main response and one for the hurdle parameter. The response variable in the model is chemiluminescence. Again, it's got a very, don't misspell that. And the we want to know if you go back to the data, right, we want to know, is the difference between vaccinated and non-vaccinated, is there a difference in the response, and is that differ by antigen? So that means we're going to put vaccination and antigen as the and their interaction, right, because there's an asterisk, as the fixed effects. And each individual chicken, right, their same blood was used for multiple antigens. So we have to use a random intercept for chicken because maybe some chickens just in general are better responders to the vaccine than others. So we account for that by putting a random intercept on chicken ID. And then the name of the hurdle parameter in BRMS notation is HU for hurdle. And I'm just going to put fixed effects on the hurdle parameter. I'm going to say this, the probability of a 0 is constant across chickens, again, because it's a very small data set with only you know, I think it's got like, how many rows does it have? It's only got 80 rows, right? So it is pretty much going to be overfit if we try to put random effects on that hurdle as well. But we can at least try to put fixed effects on it. So vaccination and antigen are the fixed effects there. The data set is immunoassay, and BRMS uses specially built-in hurdle distributions. Hurdle underscore log normal is one of them. Now we have 3 link functions. Again, these are the defaults, but I'm typing them out so you can see them. Log normal uses an identity link function. It is not, yes, there's no transformation that needs to be done on the data because log normal can take any positive value. We don't need to convert the data into another, convert the response into a different scale. So we can use an identity link function, which means there's no change. Sigma, that's like the standard deviation of the log normal distribution. It has a log link function, and the hurdle parameter has a logit link function by default. Again, because it's a probability, so we're using a log odds transformation on it, or a log odds link function. And yes, I've put priors with a mean of 0 and a small standard deviation on the fixed effects for the for both the overall mean response and for the hurdle parameter. We can put HU there, and so we're sort of being conservative on that, and similar defaults for everything else. And now I can run this. And here I'm doing the posterior predictive check plot, but I've had to add an extra scale to the plot, which is called a pseudo log scale, which allows me to plot a log transform data that includes zeros. And so we can see how well the model fits the data. Does it account for the shape of the data as well as how many zeros there are in it? Hit Control Enter to run this. So you can see that it is doing a pretty good job. Here's one peak in the data for all the zeros, and then here's another peak basically for all the immune responses that were not 0. And you can see that this is a very good fit. The model is right in between the different, or I'm sorry, the data are right in between the different draws from the posterior fitted values. So we are happy with that. Yes, so now that you've kind of seen how these different mixture models, zero-inflated and hurdle models are fit, I didn't go through as much proof of concept stuff. I've just gone ahead, and you can now see how can we estimate and compare the marginal means. Well, let's say we wanted to compare the difference between vaccinated and unvaccinated birds within each antigen. We can do that by doing EM means vaccination type antigen, and we'll use EPRED equals true, and this will give us, right, I'm not going to separate out the conditional mean and the hurdle again. I'm going to combine them into that overall response, which accounts for the zeros. Last time I showed you all of them. This time I'm just going to show you the overall response because that's usually what people are concerned with. Control Enter. And so it gives us 8 different responses. There are the non-vaccinated and vaccinated means for each antigen. So you can see that there's the non-vaccinated is lower than the vaccinated in all cases, but in some cases the magnitude of the difference is much greater. And I'm going to do contrast and I'm going to do reverse pairwise because that will give us vaccinated minus non-vaccinated, which will be a positive number. It's easier to interpret. So usually that's how I would do it if the second value is usually higher than the first. I would do reverse pairwise. Although the interpretation is the same either way. It's just easier to talk about positive numbers than negative, I think. And so these are the estimate of the difference in the response values between vaccinated and non-vaccinated for each of the antigens. And we can do a described posterior to get the Bayesian p-value on each one, just like we did before. Here the null is 0 because we're actually subtracting one mean from the other. And so we're testing the null hypothesis that those differences are 0. And we can hit Control Enter. And here we see, yes, so for some of the antigens, we have more evidence that there's a non-zero difference, that there actually is an effect of vaccinating, and for others there's not. Like this one, the 95% credible interval has a lot of probability mass near 0, and the probability map p-value is high, and that's one. Let's go back to the if you go back to the data, yes, it's this one here. So you can see that the vaccine is not stimulating that much more production of antibodies that bind to this antigen than without the vaccine, because there's already a decent amount of antibodies for that antigen, even if the chickens did not have a vaccine. It only went up a little bit after the vaccine. So there's not evidence that that's much of an increase, but for the other ones there is. And here's again in the slides I have some text for explaining how we made this figure, or what the methods were and what the results were. But here's a figure you could use. And again, I've put the data on the plot to go with the modeled mean and credible interval. So you can see A, there's some variability, and B, the model's a pretty good fit to the data. The last point I wanted to make about this is that this is a case where priors actually make a pretty big difference, because in this case, right, for for 2 of these antigens at least, there's all zeros in the data, right? There's no variability whatsoever in the data. There's no immune response whatsoever in non-vaccinated birds. All 10 tested showed 0. So because there's not very much information in the data about, we don't know what really the variability is, because the data have 0 variability. We don't know what the true mean is. Is it 0.1? Is it 0.001? We don't know because there's no variability in the data. So the priors are actually doing a lot of heavy lifting here in terms of when we estimate the difference between vaccinated and non-vaccinated. It's actually kind of sensitive to the prior, because we didn't have a lot of information in the data for this particular treatment combination. So for the exercise, I'm not going to do it here, but if you look at the exercises, you'll see there's an exercise where you can try different priors and see what the impact is on the inference, which is actually kind of interesting, I think, because then you can, yeah, I think in this case in particular, I would be especially careful when you're presenting this data to make sure you specify what prior you used and if it's sensitive, if your results are sensitive to the prior. Again, I think this is just priors or no priors. This is just why I always think it is not good to draw too much quantitative conclusions from a very small sample size study, right? There's 10 chickens in each treatment group. If we really want to get a good estimate of the true magnitude of the immune response, yes, this is enough data to probably say there is a response of some size, but to really get a good estimate of exactly quantifying its magnitude, clearly we don't have enough data to do that if we have all zeros in one group. It's not the event is too rare for us to get a very precise statistical estimate of it with a small data set. But I think that's okay, because qualitatively you can still say, well, there's, some response in vaccinated and there's no response in non-vaccinated. You don't really need statistics to show that some is bigger than none, right? So that's yeah, I think it's okay to not necessarily get too fancy with the statistics there and just say we know there's a response, but we don't really have the data to precisely estimate its magnitude. I think that's fine, and hopefully a journal reviewer would also think so. Yeah, that's I do have another example in this lesson, but to be honest with you, I don't think that I really need to go through it anyway, because it's just another example of another type of zero-inflated model for beta distribution data that has exactly 0 and exactly 1. But now that you've been exposed to other zero-inflated models, it should actually be pretty straightforward for you to apply what you've learned to that kind of model. So to be honest with you, I think you would get the most out of it if, instead of me talking to you about it, you went in and tried to run the code yourself and see what it does, because you should have a decent understanding of how these kind of models work. So I think I won't do that one next time. I'll just move to the next lesson next week. So I'd like to stop there and see what questions or comments anyone has, at least the small number of you that have stuck it out this long. Does anyone have any questions or comments?
Sorry, just a logistical question. How
Please.
long after the course concludes are you going to leave the SciNet project up, as well as like the
yes.
website and stuff?
The website indefinitely. The SciNet project, I'm not sure, because I haven't done a workshop like this on SciNet before, but I believe that after the workshops, the SciNet office usually encourages the people that run the workshops to, like, because you're added to this SEA training account only for the purpose of the workshop. But after this, I'm supposed to delete the users from that group, so you won't be able to access that shared space anymore.
Okay.
That won't matter
okay.
too much. Right. And
I
then
can still
Marina, yes,
replicate.
also
I
made
can
a
still
comment
spin up
that
the server and replicate the R project.
you could,
if
yes,
I
just
want
run
to.
it.
Okay.
If you have your own SciNet project, you have to run, once you're not on the SEA training account anymore, you have to have a SciNet project to run jobs. So if
Okay.
you don't, but you can request one using a form and get one real quick. So that's what I
Okay.
would say. Just go ahead and do that. And Marina also makes a good point that all the example data that you copy to your own space is on a folder called 90-day data, which, as the name indicates,
Yeah.
deletes itself after 90 days. So if you want to use it longer than that, copy it to a permanent space on SciNet, for sure. So yeah.
Okay. Cool.
So
All
A,
right.
you're going to
Thanks.
need to do that, and B, you're going to need to. I'm going to have to delete you from the SEA training group. So you're just going to have to run jobs on your own SciNet project. Yeah.
Gotcha. Okay. Thanks.
Those are the things to keep in mind. Thank you for asking. I should probably say that to everybody next week as well. Anybody else?
What would you do if you were, like, if you had a maximum you could observe for the Kennedy luminescence? Like, you had a bunch of points that are at, like, or this is 40,000.
I'm sorry. Say I didn't quite hear what you said.
So what if there was a maximum you could detect with the assay?
I
Like,
see.
you
So
have a
if there's
bunch
an
of points
upper
that are
bound
at
or like a,
yeah.
I see. Yeah. So then, like, if you observe that upper bound, you don't know if that's really the value or if it's actually something higher.
Yeah,
So
right.
then, yes. So then there is a thing you could do called censoring, where basically you can flag that data point as being, if it's like a million, you could say that's actually just the lower bound, and it could be higher than a million. And the uncertainty and because Paul Berkner is amazing and has put every like thing you might want in a statistical model into BRMS. BRMS has syntax for that. And so you could actually incorporate the uncertainty due to not knowing the true value of that data point into the model. And that works great if you have relatively few data points like that. But if you can imagine, like if a large percentage of your data set is censored like that, it just blows up the uncertainty because like you really don't know, like if you all you can know is that it's at least 1 million, like who knows what the real number is, right? So like if a large percentage of your data set is like that, you're not going to be able to like get good statistical estimates, even though yes, BRMS can deal with it. Yeah, it's called censoring. Like CENS is the name of the like function that deals with it in BRMS. So you should look into that. Good question, though. Okay, I am going to
So-
stop the. Oh yes, Alan, go ahead.
Yeah, no, that's okay. So, as you know, I was working with insect feeding data, and I came up with a way to sort of backwards transform it. But
That's right. Yes,
in
I
a lot
remember
of
that.
in a lot of my feeding data, what the expected value is actually a zero. And
Yeah.
what we're really
Yeah.
what we're in a convoluted way looking for, we want to hold to that zero, which means we're getting a good tight fit of our prediction that the insect is a specialist. But usually
Okay.
in really close relatives, you'll get some
Yes,
feeding.
I
So
see.
is that going
Yes,
to
proving
follow
a
that same
negative is
negative? Yeah,
proving a negative is very hard, right? Like the best you can
yeah,
do is if you see the insect feeding on that plant once, you like know for certain that it at least sometimes does it. But if you have 1,000 insects and none of them feed on it, you don't know for certain that it never does, right? So like it's just a matter
right,
of what is how much like data do you need to convince yourself that it like is
right,
negligible amount, right? So that's
right.
yeah, again,
And
that's where
would that be a zero-inflated Poisson approach? I mean, that's sort of what I played with when I transformed. I used that as part of my transformation. So I used Bayesian approaches to develop the
Yes, I
transformation,
remember
if
it.
you'll
Yeah.
recall. But you, of course, said, well, just why not just run the Bayesian models instead of doing that? Um, but so I was curious as we were working through our examples today. Obviously, it was near and dear to what I was concerned about,
Right.
but
Right.
whether an approach similar to the inflated zero Poisson would be sort of what I would use.
Yes, I think so. I can't recall exactly what that data set from when we worked on it, or I guess I didn't really work on it. You just like I just kind of like we talked about it. I don't remember actually seeing, maybe I never saw the data, but yeah, I think so. Yeah, I can't give you a good answer off the top of my head, but if you want to revisit that, I'm happy to like sit down and think about it some more. I'm not sure. I
Yeah.
think these ways of dealing with it could be good, but let's talk about it.
Okay, yeah, absolutely. We can do it offline later. That's
Yes, for
be
sure.
fine.
I just like, yeah, I can't.
But
I think
that's
you're right. You're on the right track. So yeah,
okay. Okay, very good. All right, thank you. Appreciate it.
you're welcome. Well, I'm going to go ahead and stop the recording here, but I will stick around for a couple more minutes if people still have questions. Otherwise, I'll see you next time. Thank you for being here.